Hello everyone, today I'm going to discuss with you Edexcel International GCSE 9-1 Biology Paper Topic 2. This is one of the questions in past years. Alright, this question is about biological molecule. It is about enzyme. Alright, let us see. You are given a graph. The graph shows the time taken for samples of blood to clot at different temperatures. This is the time taken for blood to clot in seconds, and this is the temperatures. Question A. When blood clots, an enzyme converts a soluble plasma protein into an insoluble plasma protein. Use the information in the graph to suggest the optimum temperature for this enzyme. How would you detect the optimum temperature for the enzyme? Yeah, alright, so the optimum temperature is the temperature where enzyme can work the best. It means that it will require the shortest time for it to convert the soluble plasma protein into an insoluble plasma protein. So from the graph, you can see, right? Okay, so this is the part, alright, where the time taken is the shortest, or specifically here. So here is actually where it is 37 degrees Celsius, alright, 37 degrees Celsius. Okay, and then the next part, what you need to do is to explain why blood takes longer to clot at 20 degrees Celsius than it does at 30 degrees Celsius. Alright, you can see that. Okay, why blood takes longer to clot at 20 degrees Celsius. So at 20 degrees Celsius, it takes about 27 seconds, while at 30 degrees Celsius, it takes about 21 seconds only. How can you answer this question? Okay, the first thing you can say that is that 20 degrees Celsius is further from the optimum temperature. In this case, the optimum temperature is 37 degrees. That is what we got from our answer in the first one. Alright, besides that, when it is further from the optimum temperature, it also means that, okay, what we can say here is that here there will be lesser movement due to lesser or low kinetic energy. Low kinetic energy. When there's low kinetic energy, okay, what will happen is that there will be fewer collision. Okay, there will be few collisions. Okay, and fewer, which causes fewer enzyme. Okay, fewer enzyme substrate complexes form. Okay, so that is what we can say of what happens here. Alright, okay, the first one you can say that, okay, you can give 2 points. These 2 points are really good enough for you. Alright, so you can mention that at 20 degrees Celsius, which is further from the optimum temperature, the enzyme and the substrate will have low kinetic energy. This causes few enzyme substrate complexes to be formed. Okay, that is for the second part. We move to the next one. Okay, and the next one is asking, use your knowledge of enzyme to explain why blood is slow to clot at 45 degrees Celsius. Alright, at 45 degrees Celsius, you can see here, Okay, the time needed for the blood to clot is much longer compared to at the optimum temperature. Okay, after the optimum temperature, usually what will happen to the enzyme is the enzyme will denature. Alright, you can mention that the enzyme will denature. Okay, the enzyme will denature. What happens if an enzyme denature is that, okay, the active site at the enzyme, okay, the active site of the enzyme will no longer, okay, active site of the enzyme, sorry of the mistakes here, active site of the enzyme will no longer feed substrate. Okay, so this is what actually happens when an enzyme denature. Okay, so you can take this point to be included for your answer. And then last part of the question, the coronary artery supplies blood to heart muscles. A heart attack may occur if the coronary artery is blocked by a blood clot. Suggest what happens in heart muscle cells when coronary artery is blocked. So what will happen if our coronary artery is blocked? We should understand the first thing is that this artery supplies us with blood. So if the artery is blocked, it means there will be less supply of blood. That is the first one. Okay, there will be less supply of blood. And when there is a less supply of blood, there is another consequence to that also. Okay, we will have less supply of glucose. Why is that so? Because as we know, okay, blood carries oxygen. Alright, and blood also. If there is less supply, I mean we cannot, oxygen is needed for respiration, and when there is no respiration, that means there will be less, we cannot respire aerobically. Okay, there will be less aerobic respiration, or you can say that you will respire anaerobically. Okay, you respire anaerobically instead. Okay, it means that when you respire anaerobically, what will happen is that there will be an accumulation of lactic acid. Lactic acid can be produced, right? And you also know that lactic acid can actually lower the pH, okay, in our blood, alright, which will, when there is a change in the pH, this will inhibit enzymes, or it will affect enzymes from functioning. Okay, so that's it. Okay, this is what will happen if our coronary artery is blocked. Thank you.